Pulsed electromagnetic field is an electromagnetic radio wave that is used to influence wound gap closure and treatment. For several decades, pulsed electromagnetic field therapy has been recognized as a type of electrotherapy that aims to treat a damaged area of tissue on a subject. It is a non-invasive, non-thermal procedure that is used to improve the structural and functional effects in both fields of human and veterinary medicine. The researchers have decided to focus on the effects of Schumann resonances on the human body. In particular, the study is based on the effects of Schumann's resonant frequencies and exposure time in the growth of normal human dermal fibroblast cells under low-intensity conditions. This study aims to present a non-invasive approach that might provide additional insight on possible mechanisms behind the cell-to-PEMF interaction within Schumann's resonant frequencies. To begin the study, NHDF cells were cultured and incubated at 4 degrees Celsius for several days. The cells were monitored and incubated until they reached 90 to 100% confluence. The cultured NHDF cells were mainly divided into an exposed group and a control group labeled as Program 1. The exposed group was further divided into Program 2, Program 3, and Program 4, where each of the exposed groups were assigned different sets of frequencies using a desktop computer connected to the MOPA or Master Oscillator Power Amplifier. All exposed programs consisted of a 10-minute and 20-minute replicate group. All programs were monitored during incubation periods at 0 hour, 48 hours, and 72 hours. A software called ImageJ was used to count the cells after each incubation period. Within the ImageJ software is the MRI wound healing tool that was used to process each well plate image to analyze the progress of the wound gap closure. To calculate the area percentage of the wound gap closure for the 48 and 72-hour incubation periods, the researchers used the area percentage calculation equation. The one-way analysis of variance test was used to determine if there were significant differences on the cell count of exposed groups as compared to the control group.
Since the cell count and the wound gap closure were closely monitored throughout the study, several tools were utilized to test if Schumann resonant frequencies affect changes on the NHDF cells. The first tool that was utilized was the ImageJ software, which counts the number of cells. The data extracted from counting the cells were then tested using one-way ANOVA through pH-stat to see which of the frequencies with the corresponding exposure times yield significant differences over the control group P1. As you can see from the table, the figures in yellow are the program groups and the exposure times that were proven to have significant differences at 95% accuracy. To break down the data, at 48-hour incubation period, P2 10 minutes was proven to have a significant difference in cell count relative to the control group P1. At the end of the incubation period, P2 10 minute, P2 20 minute, and P3 20 minutes were proven to have a significant amount of cells compared to P1. On the other hand, for the wound gap closure, the M tool of the ImageJ software was used to measure the distance of the wound edges introduced by the P200 pipette tip. Over the course of the study, it was observed that all exposed groups influenced the closure of the wound gap at 84 to 100% rate. As you can see, only P4 10 minutes was observed to be less effective in influencing NHDF cells to close the gap. One possible reason behind this could be attributed to the manual introduction of the wound on the confluent monolayer cells. Since the wound was manually induced, the sizes of the gap varies from one another. At the end of the study, it was proven that the frequencies for P2 exposed for 10 minutes can influence the cells effectively in closing the wound gap and increasing the cell count. Following this are the groups P2 and P3 for both 20-minute exposure times.